Welcome to the deep dive. Okay, today we're digging into clinical microbiology. We've got a stack of sources, and we're really aiming to distill the crucial insights for you.
Yeah, and we're not just looking at the techniques in isolation.
Right. We want to frame this with a sense of urgency, so we're using the 2014-2016 West Africa Ebola epidemic as a kind of case study. It really highlights why this field is so critical.
Absolutely. That outbreak, it was a wake-up call.
Yeah.
Unlike previous Ebola scares that were mostly rural, this one hit major cities hard. Guinea, Liberia, Sierra Leone, their health systems were just overwhelmed.
Yeah, the sources really paint a picture of how the initial response was, well, the word used is hobbled.
That's a good way to put it.
It was just this convergence of problems.
Yeah.
No readily available diagnostic tests, not enough trained people on the ground.
And real gaps in understanding basic biosafety, plus the supply chain for things like PPE, personal protective equipment just wasn't there.
A total crisis. And it forced this almost desperate innovation, didn't it? They had to figure out reliable testing while the epidemic was raging.
Exactly. It was like that phrase, building a boat while already in the water.
Yeah.
Incredibly difficult circumstances, developing and rolling out high-quality diagnostics under that kind of pressure.
But they managed it, and it really underscores how powerful molecular diagnostics have become. They got assays working that could amplify the viral RNA. But the sources suggest what really sped things up later was the approval of those simple lateral flow assays, the rapid tests.
Like a pregnancy test strip, basically, but adapted to capture the Ebola antigen. Cheap, fast, field deployable. Game-changing in that context.
Yeah.
And all that coordination, the CDC, the WHO setting up field labs, standardizing training, it all comes back to the two core missions of any clinical microbiology lab we need to focus on today.
Which are?
1, identifying pathogens quickly and accurately.
Yeah.
And 2, keeping the lab workers safe through proper containment. That's fundamental.
Okay, let's unpack this then. And starting with safety seems like the only place to begin. It's non-negotiable.
Absolutely. So the clinical microbiologist, their main job day to day is that rapid ID and, critically important, figuring out antimicrobial susceptibility. What drugs will work.
Right.
But none of that matters if the lab itself isn't safe. Safety protocols come first, before you even uncap a sample.
It's wild to think how far we've come, though. The sources mention things like mouth pipetting and smoking, eating in the lab. Were those really common practices into the 60s?
Oh, yes. It seems unbelievable now, but well into the 1960s. It took pioneers, people like Dr. Arnold Wadham at Fort Detrick, to really push for change.
Fort Detrick, right?
In the 50s and 60s, he was laying the groundwork for physical containment strategies. Before that, imagine someone mouth pipetting a bacterial culture, maybe tapping out a cigarette over the bench. Wadham's work was foundational for modern biosafety.
And that work eventually fed into the key safety manual, right, the BMBL.
Exactly. The Biosafety in Microbiological and Biomedical Laboratories, or BMBL. Its development was really spurred on by international talks in the 70s. There were growing concerns about recombinant DNA technology.
Ah, the genetic engineering worries.
Yeah, that, and also how to safely handle and study viruses known to cause cancer. So safety standards became paramount.
So how does a lab decide what level of safety is needed for a particular sample?
It comes down to a risk assessment. Basically 3 factors. What specific procedure are you doing? How experienced are the people doing it? And crucially, what's the risk group, or RG, of the infectious agent itself?
Okay, risk groups. I think I remember these. RG1 to RG4.
That's right. They rank the danger level. RG1 is low risk, things not known to cause disease in healthy adults. RG4 is the really dangerous stuff, like Ebola or Marburg virus.
And the ranking depends on things like how easily does it cause disease? How is it transmitted? Do we have vaccines or treatments?
Particularly pathogenicity, virulence, transmission route, availability of prophylaxis or therapy, all factored in.
And that risk group then tells you the biosafety level, the BSL you need.
Exactly. BSL-1 through BSL-4. Each level dictates specific requirements, lab practices, safety equipment like biosafety cabinets, and even facility design like air handling systems.
Right.
And it's important to remember, even at BSL-1, the baseline, everyone must follow standard microbiological practices. Good hand hygiene, no eating or drinking, careful handling of sharps. The basics are always required.
And then there's the gear. Everyone sees the PPE.
Personal protective equipment, lab coats, gloves, eye protection. Its job is simple. Create a barrier. block pathogens from getting to your skin, your eyes, your mouth, the entry portals.
And this isn't just good lab practice, it's backed by law, right? Things like the Bloodborne Pathogens Act.
Yep, Bloodborne Pathogens Act of 1991, Bioterrorism Preparedness Act of 2002.
Yeah.
These regulations mandate specific safety practices, including PPE use.
Okay, so safety's covered. Now, the clock starts ticking. Identification. How do labs move fast enough, especially thinking back to that Ebola crisis? It starts with the sample itself, doesn't it?
It absolutely does. The quality of the clinical specimen is paramount. We sort of mentally divide samples into two types.
Okay.
First, those from normally sterile sites in the body, think blood, spinal fluid, joint fluid. Any microbe found there is usually significant. It shouldn't be there.
Makes sense. And the second type?
Samples from non-sterile sites, things like stool samples, throat swabs, sputum. These sites normally have a whole community of microbes, the normal microbiota.
So the challenge there is finding the bad guy, the pathogen, hiding amongst all the regular inhabitants, picking it out from the noise.
Exactly. It's real detective work. And that requires getting a good specimen in the first place.
What makes a specimen good?
Well, the sources list about 6 key rules. It needs to actually represent the infected area. You need enough material. You have to avoid contaminating it with skin bacteria or whatever on the way out. It needs to get to the lab quickly, be labeled properly, and this is a big one. Ideally, you collect it before the patient starts taking antibiotics.
Ah, because the drugs might kill the bug before you can grow it.
Precisely. Makes our job much harder, sometimes impossible.
So sample in hand, what's the typical workflow?
Traditionally, it involves direct methods. First, things like looking at it under a microscope after staining or trying to culture it. Then maybe indirect methods like looking for antibodies or amplifying its genes.
Let's talk about culture first. Growing bacteria or fungi on agar plates.
Right. We use selective media stuff that inhibits unwanted microbes and differential media stuff that makes different microbes look distinct, maybe change color. It gives initial clues.
But culturing takes time, right? Days, sometimes. How do you speed that up when time is critical? Are there shortcuts?
There are. And this is where some clever tech comes in. One example is immunomagnetic bead technology, IMBs.
Immunomagnetic beads. Sounds cool. How does that work?
Okay, so these are tiny microscopic beads coated with very specific antibodies. Antibodies designed to stick only to the pathogen you're looking for.
Like little targeted magnets?
Sort of. You mix these beads into the patient sample. Let's say it's a complex mix like stool. The antibody-coated beads grab on to the target bacteria or virus. Then you use a powerful magnet on the outside of the tube.
Ah, and it pulls the bees with the captured pathogens stuck to them right out of the messy sample.
Exactly. It physically isolates your target microbe much, much faster than waiting for it to hopefully outgrow everything else in a traditional enrichment broth. Cuts the time to get a pure sample for testing down from potentially days to just hours. It's a huge advantage.
Okay, that's smart. So once you have a pure culture, maybe via IMBs or traditional methods, how do you do it?
Often with biochemical tests, we see what the microbe can metabolize, what enzymes it produces.
Yeah.
There are convenient kits for this, like the API 20E system, which is pretty classic for identifying certain gram-negative bacteria.
API 20E, how does that work?
It's basically a plastic strip with 20 little microtubes, or wells. Each well contains a different dried substrate.
Yeah.
You suspend your pure bacteria in saline and use that to rehydrate and inoculate all 20 wells at once. After incubation, say 18-24 hours, you look for color changes or other reactions in each well, positive or negative for each test. That pattern of 20 results creates a numerical code, like a 7 or a 9-digit number.
A biochemical fingerprint.
Exactly. And you plug that code into a database, and it tells you, with high probability, this profile matches Escherichia coli, or this matches Klebsiella pneumonia. Very efficient.
And identification goes hand in hand with testing susceptibility, right? Finding out which antibiotics will work.
Always. Knowing the bug's name is only half the battle. You need to know what kills it. The gold standard method is agar dilution, but it's labor-intensive.
So what's used more routinely?
Automated systems doing microbroth dilution are very common now. They give a minimum inhibitory concentration, or MIC. Also manual tests like the E-test, which is a strip with an antibiotic gradient, or the classic Kirby-Bauer disc diffusion test on an agar plate. These give you results relatively quickly, often within hours after getting the pure culture.
What about things that aren't bacteria? Fungi, parasites?
Good question. Fungi can be slow growers. Culturing them might take days or even weeks, and the media often needs antibiotics added to stop bacteria from overgrowing the plate, and maybe cyclohexamide to inhibit common environmental molds that aren't clinically significant.
Yeah.
Parasites like protozoa or helminth worms are usually identified differently, typically by microscopy looking for their eggs or cysts or the active trophozoite forms in fecal smears or other samples. Morphology is key there.
Right, just looking at their shape and features.
Exactly.
Okay, let's talk visualization more. Even basic microscopy is powerful, right? You mentioned staining.
Hugely powerful. A simple gram stain is often the first piece of actionable information the lab sends back. Just knowing gram-positive cocci in pairs and chains versus gram-negative rods immediately helps the clinician choose a likely effective initial antibiotic while waiting for the full ID and susceptibility.
So it guides empiric therapy.
Precisely. And for fungi, there are specific stains, like calcofluor white. It binds to chitin in the fungal cell wall and fluoresces brightly under UV light, making fungi pop out visually.
Now, moving beyond basic stains, you can use the immune system itself for visualization, right?
Yeah.
Antibodies.
Yes, and this is where we get really specific identification, often down to the species level. Immunofluorescence is a major technique here.
Okay, break that down. How does it work?
It leverages antibodies that have been tagged with a fluorescent molecule, a fluorochrome. There are two main ways we use it.
2 ways. Okay.
First is direct immunofluorescence. Here you're looking for the pathogen itself, the antigen. You take the patient's specimen, fix it to a slide, and add these fluorescently labeled antibodies that are specific for the microbe you suspect.
So if the bug is there, the glowing antibodies stick to it.
Exactly. You wash away any unbound antibody, look under a fluorescence microscope, and if you see glowing bacteria or viruses, boom, positive ID. You're directly detecting the antigen.
Got it. What's the second way?
That's indirect immunofluorescence. This time, you're not looking for the bug itself. You're looking for the patient's antibody response to the bug.
So detecting if the patient has been exposed.
Right. For this, you start with a slide that has a known antigen fixed onto it, say, purified virus particles. Then you add the patient's serum. If the patient has made antibodies against that virus, their antibodies will bind to the antigen on the slide.
Okay, but those patient antibodies aren't fluorescent.
Correct. So step 3 is adding a secondary antibody. The secondary antibody is fluorescently labeled, and it's designed to bind specifically to human antibodies.
Ah, so it sticks to the patient's antibodies, which are already stuck to the known antigen. A sandwich?
Kind of a layer, yeah. So if the patient's antibodies were present and bound in step 2, the fluorescent secondary antibody will bind to them in step 3, and the whole complex will glow. Fluorescence means the patient has this specific antibodies you were testing for.
Indirect because you're detecting the patient's response, not the bug directly. But what about things too small for even fluorescence microscopy, like viruses? Or when you just need answers fast, like in the Ebola outbreak?
That's where molecular methods really shine. PCR polymerase chain reaction and related techniques are now routine, especially for viruses. They provide the speed that was so desperately needed.
The star player seems to be real-time PCR.
Yeah, real-time PCR or qPCR is fantastic. It's rapid, highly accurate, and it's quantitative. It doesn't just tell you if the pathogen's genetic material is there, it tells you how much is there, which can be really important for monitoring treatment or infection severity.
And for RNA viruses like Ebola or HIV or flu, you need an extra step first.
Right. PCR amplifies DNA, so for RNA viruses, you first have to use an enzyme called reverse transcriptase to convert the viral RNA into DNA. Then you run the PCR. That's called RT-PCR, or reverse transcriptase PCR.
Okay, but the really mind-blowing version seems to be multiplex PCR.
Oh, it's incredibly powerful. Multiplexing means you put multiple different sets of PCR primers into the same reaction tube with the patient sample.
So you can test for lots of things at once.
Exactly. Instead of running separate PCRs for flu A, flu B, RSV, coronavirus, etc., you can run one single test that screens for, say, 20 different respiratory viruses simultaneously. Or panels for bloodstream infections that detect dozens of bacteria, fungi, and key antibiotic resistance genes, all from one blood draw.
Wow. That completely transforms how quickly you can get targeted treatment started. Hours instead of days.
Absolutely game-changing for patient outcomes. That speed is critical.
And for bacteria specifically, there are other molecular ID methods too.
Yes, for really fine-tuned identification or tracking outbreaks, we use things like ribotyping. This involves sequencing the gene for the 16S ribosomal RNA.
Why that gene?
It's highly conserved across bacteria, meaning it changes very slowly over evolution, but it has enough variation to distinguish between different genera and species. It's like a bacterial barcode.
And MLST?
Multi-locus sequence typing.
Yeah.
This goes a step further and sequences fragments of several different housekeeping genes, essential genes found in all strains. The combination of sequences across these multiple genes gives a really detailed genetic fingerprint or sequence type, ST. This is invaluable for epidemiology, for tracking exactly how a specific pathogenic strain is spreading through a population during an outbreak.
Okay, shifting gears slightly. Let's talk about serology, looking for antibodies again, but maybe using different techniques.
Right. Serology, that indirect detection via antibodies, is crucial when you can't easily culture the microbe. Maybe it's too dangerous, like some BSL-3 or 4 agents, or requires specialized techniques like Treponema pallidum, the syphilis bacterium,
Or HIV.
or HIV exactly. Another key time is if the patient has already started antibiotics, which might wipe out the bacteria, making culture useless, but the antibodies will still be there.
And serology gives you that time element too, right? IgM versus IgG.
Yes, that's a really important concept. IgM antibodies are typically the first type produced during a new active infection. They rise quickly and then usually decline. Ig antibodies rise a bit later but tend to persist much longer, sometimes for life, indicating a past infection or immunity from vaccination.
So looking at the levels or the ratio of IgM to IgG against a specific pathogen can tell you if the infection is recent and active or if it happened weeks or months ago, like a laboratory time machine.
That's a great way to put it. It helps stage the infection.
What are some common serological test formats?
Well, the simplest are probably agglutination reactions. This is just mixing the patient's serum, which might contain antibodies, with particles or cells that have the corresponding antigen on their surface. If the antibodies are present, they'll cross-link these particles, causing them to clump together visibly. That clumping is called agglutination.
Okay, like the rapid plasma reagent test for syphilis screening.
Exactly. Or latex agglutination tests, where tiny latex beads are coated with either antigen or antibody, and you look for clumping when mixed with the patient sample. Quick and easy to see.
But then things get a bit weird. The sources talk about some tests where a lack of reaction is the positive result, like viral hemagglutination inhibition.
Ah, yes, those are clever but definitely counterintuitive. So some viruses, like influenza, naturally cause red blood cells, RBCs, to clump together. That's called hemagglutination.
Okay, virus plus RBCs equals clumping.
Right. Now, in the hemagglutination inhibition test, you first mix the patient's serum with the virus. If the patient has neutralizing antibodies against that virus, those antibodies will bind to the virus particles and block their ability to clump the RBCs.
So antibodies block the virus.
Then you add the RBCs. If the antibodies were present and neutralized the virus, the RBCs don't clump. They just settle to the bottom of the well. So no clumping equals a positive result, meaning the patient had the specific antibodies.
Wow, okay. Lack of clumping means success. That takes a minute to wrap your head around.
It does. And complement fixation is similar in that way. It's another very sensitive but also complex two-stage assay.
How does that one work?
Okay, stage 1. You mix a known antigen, the patient's serum, which might have antibodies, and a carefully measured amount of complement, that's a set of proteins involved in the immune response.
Right.
If the patient's serum does contain specific antibodies, they'll bind to the antigen, forming immune complexes. These complexes then activate and fix or consume the complement proteins you added. All the complement gets used up.
Okay, so if antibodies are present, complement disappears.
Exactly. Now, stage 2, you add an indicator system. This is usually sheep red blood cells that have been pre-sensitized with antibody. Normally, complement would lyse or destroy these indicator cells.
Right.
But if the patient's antibodies were present in stage 1 and already used up all the complement, there's no complement left to lyse the indicator cells in stage 2. The sheep RBCs remain intact.
So again, a lack of reaction, no lysis of the indicator cells is the positive result, indicating the patient's specific antibodies were present initially.
Precisely. No lysis equals positive test. Success is seeing nothing happen in the final step. Very sensitive, but tricky to perform correctly.
Definitely counterintuitive. Are there more straightforward tests used commonly now?
Oh, absolutely. The most widely used serological method today is probably ELISA, the enzyme-linked immunosorbent assay. It's versatile, sensitive, and can be automated.
ELISA. How does that work? Still using antibodies?
Yep. Usually involves antibodies linked to an enzyme. When the right substrate is added, the enzyme causes a color change you can measure. There are a couple main types.
Okay.
Indirect ELISA is common for detecting patient antibodies. You coat the wells of a microtiter plate with the known antigen, add patient serum, wash, then add an enzyme-linked secondary antibody that binds human antibodies. Add substrate, look for color, more patient antibody.
Similar principle to indirect immunofluorescence, but with an enzyme of color change instead of fluorescence.
Exactly. And then there's direct ELISA, sometimes called the sandwich ELISA. This detects the antigen itself.
How's that set up?
You coat the well with a capture antibody, add the patient's sample. If the antigen is there, it binds to the capture antibody. Wash. Then add a second different detection antibody that's enzyme linked and also binds to the antigen, but at a different spot.
So the antigen gets sandwiched between two antibodies?
Precisely. Capture antibody on the bottom, antigen in the middle, enzyme linked detection antibody on top. Add substrate, color develops if the antigen was present.
Got it. And this ELISA technology is what's in those rapid tests too.
Often, yes. Lateral flow assays like those rapid strep tests or the Ebola antigen test we mentioned are essentially simplified strep-based ELISAs or similar antibody-antigen interactions.
How do they work so fast?
The sample fluid containing the potential antigen wicks up a porous membrane like filter paper. As it flows past a test line, it encounters antibodies fixed there. If the antigen is present, it binds, and this captures colored particles or triggers a reaction that makes the line visible. There's also usually a control line further up to show the test strip worked correctly. Simple, fast, cheap, no equipment needed.
Brilliant. One last thing on serology, what's a titer?
Titer. It's basically a measure of how much antibody is present.
Mm-hmm.
You typically do serial dilutions of the patient's serum, 1.2 and 1.40, 1.80, and so on, and test each dilution using something like an agglutination or ELISA test.
Okay.
The titer is reported as the reciprocal of the highest dilution that still gives a positive result.
Mm-hmm.
So if the 1.160 dilution is positive, but the 1.320 is negative, the titer is 160. A higher titer generally means more antibody. Comparing titers over time, acute versus convalescent samples, is really useful for diagnosing active infections.
Wow. Okay, we have covered a lot of ground here. From the absolute bedrock of biosafety, thinking back to WADM and the BSLs, through culturing, the smarts of IMBs, biochemical IDs, the speed of molecular methods like multiplex PCR,
Right.
and then into the whole world of serology using antibodies for fluorescence, ELISAs, those tricky inhibition and fixation tests, and measuring titers. It's an incredible arsenal of techniques.
It really is. Yeah. And bringing it back to where we started with the Ebola crisis. All these tools, from the foundational safety protocols to the most advanced molecular and serological assays, are what stand between us and the rapid spread of dangerous pathogens. They are the essential machinery for global health security, for tracking outbreaks, for developing responses. The clinical diagnostic lab is, it's truly the front line.
Absolutely. A crucial, often unseen defense. So as we wrap up, here's something for you, our listeners, to ponder. We've seen the amazing speed and sensitivity of molecular tests like PCR. How much emphasis should labs still place on mastering traditional culture and biochemical methods, especially when the goal is often determining antibiotic susceptibility, which culture still does very well? What are the real-world trade-offs in the lab between maximum speed and maybe a more thorough traditional workup? Something to mull over.